To get a recombinant DNA, it is essential that the DNA be cut and then joined. And in gene cloning, essentially, the biological tool that helps in cutting the DNA is what we know as restriction endonucleases. So essentially, restriction endonucleases are the molecular Caesars of the gene cloning technique. So let us look at the learning outcomes of this session, which is going to focus on restriction endonucleases. There are several types of restriction endonucleases, of which restriction endonuclease 2 cuts the DNA at very specific sequences, not just with precision but also with reproducibility. It means that it is always going to cut only at those specific sequences, giving discrete fragments. In a cloning experiment, ideally, the same restriction endonuclease is used to cut the vector as well as the DNA that is to be cloned. The restriction endonucleases cut the DNA in such a way that it either gives rise to blunt ends or sticky ends, with sticky ends being preferred as they would be more useful or more fast or rapid in getting inserted into a vector, but the vector should also have the same sticky ends. Analysis of the restriction digest can be done using gel electrophoresis. So, restriction endonucleases were discovered by Warner Arbor, Hamilton Smith, and Daniel Nathans, and they won the Nobel Prize for the discovery of restriction endonucleases and its application in 1978. Now, interestingly, Warner Arbor was who actually discovered these enzymes and postulated that these enzymes bind to the DNA at very specific sequences and cut at those sequences only. Hamilton Smith verified Arbor's hypothesis by looking at what is understood to be today host control restriction, where a bacteria is able to produce this restriction endonucleases, and these restriction endonucleases cut again at very symmetrical sequences. Nathan, that is Daniel Nathans, actually made use of restriction endonucleases to study various aspects and gave rise to, or what began was, an era of genetic engineering and modern genetics. So their contribution is outstanding just because of the fact that their study on restriction endonucleases was a breakthrough to in what is understood to be recombinant DNA technology. So let us look at what is the phenomenon of host control restriction, by way of which one could understand restriction endonucleases, or the discovery basically was on the host control restriction phenomenon. So what was observed is that there were certain bacteria, okay, that would be infected with the phage. So the DNA of the phage would be injected into the bacteria, but the bacteria would have certain very specific enzymes that would bind to the phage DNA at specific sites and cleave the phage DNA, thereby stopping the multiplication of the bacteriophage within the host. So the host is restricting the multiplication of the bacteriophage, and that is why it is called as host controlled restriction. Now, here a question that comes up is that if the bacteria is synthesizing restriction endonucleases that can cut a DNA, so phage DNA, if it is able to cut the phage DNA, what about its own DNA? So the question that comes about over here is, will the restriction endonucleases that the bacteria produces will cut its own genome? The answer to that is, of course, no. So the restriction endonucleases can cut only the foreign DNA but not its own DNA. So how is this possible? So what was observed is that the bacteria is able to methylate those sequences which are specific for the restriction endonuclease that it produces. Thereby, the restriction endonuclease cannot recognize those sequences and hence will not cut the DNA. If, for example, the methylation of these sequences are not possible because of a mutation or because of a lack of a particular enzyme, then these sequences not being methylated will be cut by the restriction endonuclease. And so in that scenario, the restriction endonuclease will also cut the bacterial genome. So essentially, the understanding of restriction, that's the reason why the word restriction has come up. That is, the host bacteria on producing these endonucleases, the endonucleases are able to restrict the foreign DNA from getting multiplied in the cell because the endonuclease cuts the foreign DNA only. Now there are three types of restriction endonucleases. Type 1 is a pentameric complex, that is, it is made up of 5 subunits with one subunit specific for restriction, one specific for methylation, and the third specific for sequence recognition. So therefore, what has been observed is, as a pentameric complex, the mechanism is slightly complex. The restriction endonuclease type 2 is much simpler as it binds to a recognition sequence and its cutting is happening at the recognition sequence itself. Either it is just outside the restriction site, so the recognition sequence is also called as a restriction site, or it is within the restriction site itself. Type 3 again has a complex mechanism as it recognizes more than one recognition signal, and these recognition sequences are long range recognition sequences, and therefore the entire mechanism of cutting, it does happen, but it is much more complex. So for the simplicity of its mechanism, restriction endonuclease type 2 has found or has created its place in gene cloning experiments as it gives rise to very discrete fragments. So, is there specific sequences? So, the specific sequences which are called as restriction sequences can either be tetranucleotides or they can be pentanucleotides, hexanucleotides, octanucleotides, or they can be longer than octanucleotides. So, say for example, SAU3A, this is a restriction endonuclease that has been isolated from Staphylococcus aureus, and this specifically cuts at a tetranucleotide sequence. As you can see, it cuts a GATC, while ALU1, which has been isolated from Atrobacter luteus, cuts at AGCT. So this also is a tetranucleotide and this also is a tetranucleotide, but the sequence is different. So both the enzymes, that is the SAU3A and the ALU1, recognize a tetranucleotide sequence, but the recognition sequence is only one. Similarly, you have EcoR1, this is isolated from E. coli, that cuts a hexanucleotide. So the sequence that it recognizes is a hexanucleotide, GAATTC, and it cuts only within this sequence. Now, what has been observed is that there are certain restriction endonucleases that do have degenerate recognition sequences. What do we mean by that? We mean by this is that it can cut at more than one recognition sequence, but those recognition sequences are related to each other. So the sequence has some homology. So, say for example, HINF1, this is isolated from Haemophilus influenza, and this restriction endonuclease can cut at GAATC, which is a pentanucleotide. It can cut at GATTC, it can cut at GAGTC, and it can also cut at GACTC. So what is observed is that this third nucleotide in the sequence in all of these is varying. The remaining of the nucleotides in the sequence remains the same. So essentially, it recognizes any sequence that has GA, N, this can be any nucleotide, T and C. Therefore, these are degenerate recognition sequences for a single restriction endonuclease. Another interesting facet about the restriction site is that most of the restriction sequences, not all, but most of the restriction sequences are palindromes. What do we understand by palindromes? So when you have a sequence from 5 prime to 3 prime, say GATC, and when you read it on the other strand from the other direction, again 5 prime to 3 prime, then it will read the same. So GATC, and when you read it from this side, it is also going to be GATC. This is what a palindrome is. Let's take the example of GAATTC. When you add the complementary, you will find this is GAATTC. So again, you can see that it is a palindromic sequence. So many such sequences which are restriction sites are palindromes. Now, is there a very specific way of cutting, or it cuts always in the same way, or are there different ways of cutting? So say for example, this is the recognition sequence. Suppose the cut is made in such a way that the cut on both the strands is at the same site or the same point of the recognition sequence. Then what you would get is two a cut made with blunt ends being formed. So here you have this end and this end. So these ends are what are called as blunt ends. As you can see, ALU1, when it recognizes AGCT, it cuts at this point, and so you have a blunt end being formed in both the cases. One another way of cutting is, say you have a recognition sequence, but in this recognition sequence, the cut is made on both the strands, but it is made such a way that the cut is always between the first and the second nucleotide from 5 prime to 3 prime. First and the second nucleotide from 5 prime to 3 prime. So therefore, on cutting, what you will get is one end would be this and the other end would be this. So you would definitely have a 5 prime overhang being formed because of the staggered cutting. Now, interestingly, we know that this n, which is this n, is complementary to this n. So therefore, all these n's are complementary to each other, and therefore they would tend to anneal to each other again. And that is the reason why these staggered ends or overhangs are also called as sticky ends. They would like to anneal with each other again, and therefore they are called as sticky ends. So you can see this with EcoR1. It cuts the GAATTC in such a way that it forms a sticky end structure. So therefore, in gene cloning, sticky ends are preferred because they will immediately try to complementarily bind to each other and therefore anneal to each other. So if you have a vector which has the EcoR1 sticky end, then it would try to anneal to any DNA which has a EcoR1 sticky end. Therefore, it is always said the DNA that has to be cloned is cut by the same endonuclease as is used to cut the vector DNA. Now, there are certain restriction endonucleases which recognize different sequences, but on cutting, they give rise to the same sticky end. So you can see BAMH1 gives CTAG as a sticky end. BGL2 also gives sticky ends, which is the same. So you can see that in all the three cases, the sticky ends are same, but the recognition sequence in all the three are different, at least in the first nucleotide pair. So therefore, this is an interesting facet that has been observed. Moreover, what has been observed are there are isosceles present. What do you mean by isosceles? They are restriction endonucleases that are different, but these different restriction endonucleases recognize the same recognition site or the restriction site and also cut at the same point. So this will give the same sticky end as SPH1 gives. That is again another important fact. Then you have what is called as neosesomers. So neosesomers are again different restriction endonucleases which recognize the same restriction site, but they cut at different points in the restriction site. So, in a single DNA, one can have several restriction sites for a single endonuclease, or there can be several restriction sites for different enzymes possible, which means we are talking about one DNA having several recognition signals for not just one restriction endonuclease but many restriction endonucleases. Now, restriction digestion therefore can be done in the lab because it is a simple enzymatic activity. So what is required to carry out restriction digestion in the lab? One of course needs the DNA that has to be digested. So therefore this DNA that is chosen has to be of good quality. That means its A260 by 280 has to be close to 1.8. Why do you need a good quality DNA? Because if the quality of the DNA is not good and there is contamination of proteins, then the efficacy with which the enzyme can cut will be decreased. And therefore, to have a good quality DNA is needed for restriction digestion. Also, what is important is to know the amount of DNA that is present. Now, why is quantity important? Because an enzyme substrate binding has to do with the equivalent amount or a certain amount of substrate to be present. So, for example, if you have the RE, then how much amount of RE can cut how much amount of DNA? That is important for ensuring that it is a successful restriction digestion. Now, therefore, what is first added in a restriction digestion assay is the DNA that is to be cut. So say a certain volume has been taken. Say this volume contains 1 microgram of DNA. Now, a buffer is then added to this DNA. Why a buffer is added before the restriction endonuclease is because the buffer will precondition the assay system to enable the restriction endonuclease to work at its best. So the buffer generally tries to obviously maintain the pH, and we all know that enzymes have their optimum pH at which they are most functional. So therefore, in most cases, what has been found that a pH of around 7.4 is optimum for several restriction endonucleases. It can of course vary. So therefore, based on what restriction endonuclease is being used, the buffer will be having a pH equivalent. Then in restriction digestion, most restriction endonucleases require magnesium as a cofactor, and the buffer also has NaCl, and together MgCl2 and NaCl maintain the ionic strength that is required for the restriction endonuclease to function. So if there are difference or changes in the amount of magnesium, NaCl or pH, then the entire enzyme activity will not take place. It will be an inefficient assay system. And you will not find digestion of the DNA happening. Other than this, what is also added is dithiothriotol. Dithiothriotol is a reducing agent, and this is added to just ensure that the enzyme does not become inactive and it is present in a form that is most active for the enzyme. So therefore, a certain amount of buffer is added to the DNA that has been already added. And this is mixed well so that the DNA is now in a condition or in an environment which is conducive for the restriction endonuclease to act. Then the restriction endonuclease has to be added. Commercially available restriction endonucleases are available in such a way that the units per ml is already provided. So based on what is the amount of DNA present, one has to take that volume of restriction endonuclease that can cut the DNA. Mostly, it is 1 unit of restriction endonuclease can cut 1 microgram of DNA. So suppose you have 1 microgram of DNA, you have to take a volume of RE that contains 1 unit. So after the restriction endonuclease has been added, this is again mixed and Then water is added to make up the volume. So the assay system volume has to be made up to a certain level. Once it has been made, then it can be incubated for the restriction digestion to take place. This is obviously water and not a tris EDTA buffer for obvious reasons. So therefore, the incubation has two important facts again. One, an optimum temperature has to be maintained because we also know that enzymes need an optimum temperature to function, and appropriate time period has to be given for the reaction to take place. So it has been observed that in 1 hour's time, generally, restriction endonucleases are able to cut the DNA completely. Completely in the sense at the sites where it can bind and cut. If you keep it for less time, then it is possible that the digestion is not complete, it is only partial. So once you have the digestion done, you will find that the DNA is now present in fragments, and the fragment sizes will vary depending on where and how the restriction endonuclease has cut. Now these fragments of DNA can be used for cloning, but before it is used for cloning, it is imperative to ensure that the enzyme that is present in this is denatured. Because if you add this DNA, or if you take out an aliquot of this DNA and put it into a system which has the vector, then it is possible that the enzyme can cut the vector. So in order to ensure that the enzyme does not cut any other DNA, it has to be denatured. So one of the ways by which it can be denatured is by using phenol or by heating, or it can be inactivated by adding EDTA. So when EDTA is added, the EDTA will chelate magnesium ions, and because it chelates magnesium ions, the endonucleases will become inactive. These fragments are also analyzed, and it is important to analyze because you will know a certain amount of aspects related to restriction digestion. The analysis is generally done using gel electrophoresis. So as you can see over here, suppose you load three different samples in the gel that has been prepared. See, this as a sample that is cut by one restriction endonuclease, this is by a second restriction endonuclease, and this is by a third restriction endonuclease. So once the DNA is run and you observe it under the UV transilluminator, you will find the different fragments have been separated out as bands. So you know that cutting of this sample by this restriction endonuclease has led to formation of four fragments. Cutting of the same sample by another restriction endonuclease has led to formation of five bands. That means five fragments of DNA, and likewise. So by a simple gel electrophoretic run, what you will definitely observe is how many number of fragments have been obtained after the restriction digestion. If one runs a molecular weight marker as well, so molecular weight marker generally has fragments of known size. So depending on the size of the fragment in an electrophoretic run, you know that the smallest fragment will travel the fastest and the largest will travel the least. Based on their sizes, therefore, and their migrations will happen, and based on that, you will have the known fragments with the known molecular weights separate out. Now, when the unknown is also run along with it, visually one can make out or approximate the size of the fragments that have been obtained due to restriction endonuclease digestion. So this is basically an estimate of the molecular size. You can basically say, okay, this is about 3000 base pair or this is about 2000 base pair. But to get an accurate size, one needs to do, or one needs to basically uh, um, calculate the migrated distance. So from the well in which the sample was loaded the to the place where the band has been formed, that is the distance traveled by the DNA fragment. So that is something that has to be done for every fragment that has been isolated, and based on that you have a calibration curve of the known molecular weight markers. And once you know the migration or the distance traveled by these samples, you can basically find out the molecular size by looking at or by the graph that you get because of the known molecular weight markers. So this would basically give an accurate size, and this is generally because of the distance migrated by the sample being compared to the standard. Now, another thing that is important in gene cloning is restriction map. What do we mean by restriction map? So let's consider you have a DNA, okay? And in this DNA, there are two restriction sites for EcoR1. That is, the EcoR1 can bind over here and cut. It can bind over here and cut. So for this DNA, the restriction map for EcoR1 is this. Now, if you have HIND3 recognition sequences, one can identify where exactly HIN3 can cut. So now this DNA has a restriction map for EcoR1 as well as HIN3. Suppose you have another restriction digestion site. Okay, say SPA1 recognition sequence is this, which means what we are looking at is that this particular DNA has several recognition signals for three different restriction endonucleases, and for HIN3 it has several recognition sequences. For EcoR1 also there are more than one recognition sequences, but for SPA1 it has only one recognition sequence. So this is what a restriction map is. Okay. Now, why do we need to know the restriction map of the DNA? It is important to know the restriction map of the DNA if you have to use this DNA to clone the gene of your interest. So for example, if the gene of your interest is present over here, then you would be interested in getting a fragment that contains this gene. Or say you have a gene present over here and then you need this. Then you need to identify a fragment that will contain this gene. Or the gene is present over here accordingly. Currently, let us take that this is the gene that we are interested in. So if you cut it with EcoR1, if you cut the DNA with EcoR1, you will get this is fragment 1, this is fragment 2, and this is fragment 3. So when the linear DNA has two recognition sites for EcoR1, you get 3 fragments. Okay. Now, suppose therefore in this case, the gene that you are trying to clone will be present in fragment number 2. Now suppose you cut with HIN3. Then HIN3 will give you fragment 1 till here, fragment 2 till here, fragment 3 till here, and fragment 4. So a linear DNA that has 3 restriction sites will give rise to 4 fragments. That is n plus 1. Now say for example you cut with SPA1. If you cut with SPA1, it has only one recognition signal or sequence or restriction site. So the DNA will be cut into only 2 fragments. Now in this scenario, therefore, when you cut with EcoR1, the gene is present in fragment 2. When you cut with HIN3, the gene is present in fragment 2. When you cut with SPA1, the gene is present in fragment 1. So, of the 3 restriction endonucleases, which would you choose to get a fragment that contains the gene of your interest? We would obviously go for HIND3. Why? Because the gene is present in a fragment size that is small and hence easy to handle and clone. If it is a longer DNA that contains the gene of interest, it may become difficult to clone it because to fit it into a vector may be difficult. But if it is a small fragment, then fitting it into a vector will be easier. And so from this, you understand that if you know the restriction map, you can actually decide which restriction endonuclease you want to use to do the cutting of the DNA to get the fragment that contains the gene of your interest. So, restriction map is something that is important to find or too important to do before you actually go in for a gene cloning experiment. So let us make the conclusions. Restriction endonucleases are of three types, of which restriction endonuclease 2 is used in the cloning experiments. They recognize and cut within a specific recognition sequence, giving rise to either blunt end or sticky ends. Restriction digestion can be performed in the lab and fragments can be analyzed for further experiments. Analysis is generally done using gel electrophoresis. Knowing the restriction map of a DNA and vector can enable one to choose which restriction endonuclease to use. Thus, restriction endonucleases, the molecular Caesars in gene cloning, are able to give rise to discrete fragments of DNA which can be cloned into vectors that too can be cut by the same endonuclease. Thank you.